In the summer of 1947, a young Bedouin shepherd went looking for a lost goat near the cliffs above the Dead Sea. He tossed a stone into a dark opening in the rock, expecting silence. Instead, he heard something shatter. Inside that cave sat clay jars that had not been touched in nearly 2,000 years, and wrapped inside them were strips of ancient leather covered in Hebrew script. Nobody in that moment could have guessed that decades later, a team of geneticists would crack open the same fragments at the molecular level and discover that everything scholars thought they knew about these documents was incomplete. This is the story of how a casual conversation between two scientists turned into one of the strangest and most important discoveries in the history of biblical archaeology, and how DNA extracted from animal skin ended up rewriting the story of the most famous ancient manuscripts on Earth. The scrolls found in that cave, and in 10 others nearby over the following years, became known as the Dead Sea Scrolls. Altogether, they amount to roughly 900 separate manuscripts, broken into tens of thousands of fragments, some no bigger than a fingernail. They include the earliest known copies of Hebrew biblical texts, along with hymns, legal writings, and documents describing the rules of a religious community. Radiocarbon dating and paleography place them between the 3rd century BCE and the 1st century CE, meaning some of these pieces of parchment were already old when Roman legions were marching through Judea. For a text-obsessed civilization like ours, finding manuscripts that old, in that condition, was almost unthinkable. It rewired how historians understood the roots of both Judaism and Christianity, because the scrolls captured a snapshot of Jewish religious life during the exact era when Christianity was beginning to take shape. For 70 years, the study of these scrolls was the work of paleographers, historians, and archaeologists. They compared handwriting styles page by page, hoping to identify which scribe wrote which fragment. They examined the color and thickness of the parchment, trying to guess which torn pieces once belonged to the same sheet. They cross-referenced content, looking for words or phrases that might link one scrap of text to another sitting in a different box, in a different museum, sometimes on a different continent. It was an enormous, painstaking, and often frustrating exercise, something like assembling 1,000 jigsaw puzzles that had been dumped into the same bin, missing most of their pieces, with no box lid to show what the final picture should look like. Getting it wrong had real consequences. Join two unrelated fragments together, and you could accidentally invent a version of scripture that never existed, or attribute an idea to an ancient community that never held it. Out of all that careful, decades-long labor came a widely accepted explanation. Most scholars concluded that the scrolls belonged to a Jewish sect called the Essenes, a group mentioned by the ancient writers Josephus and Pliny the Elder. According to this theory, the Essenes lived a secluded, highly disciplined life at a settlement called Qumran near the caves, followed strict purity codes, and spent much of their time copying and preserving sacred texts. When Roman forces closed in during the Jewish revolt that began in 66 CE, the story goes, this community hid their library in the surrounding cliffs, where it waited undisturbed until a shepherd's stone found it by accident 19 centuries later. This idea explained a lot. It matched descriptions from ancient historians. It fit with the sectarian rulebooks found among the scrolls, and it accounted for a solar calendar in some texts that differed from the lunar calendar used by mainstream Jewish authorities in Jerusalem. But underneath that tidy narrative, cracks kept appearing that traditional methods could never quite patch over. Some biblical books turned up in the collection in 2 or 3 noticeably different versions, with entire passages missing, reordered, or reworded. Handwriting analysis showed dozens of different scribal hands at work, using different spelling conventions and different levels of skill. The physical parchment itself ranged from fine, carefully tanned material to rough, uneven scraps, and radiocarbon testing suggested some scrolls predated the Qumran settlement altogether. If this was really the private collection of one isolated, insular community, why would its library be bursting with contradictory copies of the same sacred texts? Why keep multiple versions of a book if your community had already settled on which version was correct? These were the kinds of questions that traditional scholarship could raise convincingly, but never fully answer, because the tools of paleography and textual comparison had reached the edge of what they could tell us. The breakthrough started, almost absurdly, over a shared academic bus ride. In 2012, Oded Richavi, a molecular biologist at Tel Aviv University who studies genetics in microscopic worms, found himself seated next to Noam Mizrahi, a scholar of ancient Hebrew literature, during an orientation event for new faculty. They got to talking, and Mizrahi described the maddening puzzle piece problem that had plagued Dead Sea Scrolls research for generations, the constant guessing about which fragments belonged together and the total absence of any objective way to test those guesses. Richavi, thinking like a geneticist, asked what the scrolls were actually made of. Mizrahi told him, parchment, made from animal skin. And Richavi realized something that should have seemed obvious in hindsight, but had never been applied to this problem before. If the scrolls were written on animal skin, then somewhere in that skin, buried and broken down by 2,000 years of decay, there might still be genetic material. Not DNA from the ancient scribes who wrote the words, but DNA from the sheep, goats, and cattle whose hides had been turned into writing surfaces. That idea sounds simple, but turning it into usable science took years of extremely careful work, and the obstacles were enormous. The Dead Sea Scrolls are treated as sacred and irreplaceable artifacts, and destructive sampling of any kind is essentially forbidden. Researchers could not cut new samples from intact fragments or drill into the parchment. Instead, the team, which eventually grew to include Richavi, Mizrahi, geneticist Matthias Jakobsen at Uppsala University in Sweden, and genomics researcher Christopher Mason at Weill Cornell Medicine, worked with the Israel Antiquities Authority to gather minuscule bits of material that had already detached from the fragments over the decades of handling, or tiny scraps that could be responsibly removed from blank, unwritten sections without touching a single letter of text. From this material, spread across dozens of separate pieces, they attempted to pull out genetic sequences old enough and clean enough to trust. The scale of degradation they were up against is hard to overstate. Making parchment in the ancient world involved soaking, scraping, and stretching animal skin, a process that damages genetic material at a chemical level even before the writing begins. Then came iron-based inks, which are corrosive to the material they sit on. Then 2,000 years sealed in desert caves, exposed to heat swings, humidity, and mineral buildup. And on top of all of that, 70 years of modern researchers, curators, and visitors physically handling the fragments, each contact potentially depositing fresh human DNA on top of the ancient material the team was trying to isolate. To have any hope of separating genuine ancient sequences from modern contamination, the researchers had to work inside specialized ancient DNA laboratories under extreme contamination controls, full protective suits, positive air pressure, UV-sterilized equipment, and rigorous cross-checking between labs to confirm that what they were sequencing was actually old and actually came from the animal, not from someone's coffee cup or fingertip decades later. The team focused mainly on mitochondrial DNA, which survives better in ancient samples because each cell carries hundreds of copies of it, giving researchers more raw material to work with even when a sample is badly degraded. Where possible, they also examined nuclear DNA, which is far scarcer in old material but carries more detailed information, including clues about how closely related different animals were to one another. After years of trial and error, sample collection, and laboratory validation, the team had usable genetic sequences from roughly 24 scroll fragments, along with a few related leather items, such as sandals and water skins, found in the same region. What they found when they finally analyzed the results, published as the cover story of the journal Cell in 2020, was more revealing than almost anyone had expected. The first finding was straightforward on its face, but carried enormous weight. Nearly all of the tested fragments were made from sheepskin, which made sense given that sheep and goats are well suited to the harsh, dry terrain around the Dead Sea. But 2 of the fragments were made from cowhide instead. That distinction matters enormously, because raising cattle requires large amounts of water and substantial grazing land, resources the Judean desert simply does not have. A community living at Qumran could not have realistically raised or regularly obtained cattle for parchment. Any fragment made from cowhide almost certainly did not originate in that desert settlement. It had to have been produced somewhere else, somewhere wetter and more fertile, and then carried into the Qumran caves later. That single biological detail, cow versus sheep, was the first piece of hard, physical evidence that not every scroll in the collection began its life at Qumran. The two cowhide fragments turned out to belong to copies of the book of Jeremiah, and this is where the story gets genuinely fascinating. Scholars had long known that the Dead Sea Scrolls preserve at least two very different versions of Jeremiah. One closely resembles the Hebrew Bible familiar today, longer and organized in a particular order. The other is noticeably shorter, missing whole sections, and arranged differently. Researchers had debated for years whether this reflected two distinct textual traditions developed in separate communities, or simply variation introduced by individual copyists. Before the DNA analysis, one of these cowhide Jeremiah fragments had actually been assumed by some scholars to belong to the very same scroll as a nearby sheepskin fragment. The genetic evidence flatly disproved that assumption. Different animals, different likely places of origin, meaning these were never pieces of a single physical scroll at all. Because the divergent Jeremiah text happened to be the one written on cowhide, brought in from outside the desert, the genetic data supported the idea that these were genuinely separate versions of the prophetic book, circulating and being copied in different Jewish communities at the same time, not just copying errors within a single tradition. Beyond species identification, the deeper value of the DNA work came from comparing fragments to each other directly. By examining shared mitochondrial and nuclear markers, the researchers could sometimes tell whether two pieces of parchment came from animals that were closely related, distantly related, or seemingly unrelated. This gave scholars something they had never had before, an objective, biological way to test whether fragments plausibly belonged to the same original document, instead of relying entirely on subjective judgments about handwriting style or the visual appearance of the leather. In several cases, this genetic fingerprinting supported connections that textual scholars had already proposed based on spelling patterns and scribal habits specific to the Qumran community, giving those earlier hypotheses real, independent confirmation for the first time. Taken together, these findings pushed researchers toward a more nuanced picture of what the Dead Sea Scrolls actually represent, rather than being the exclusive, self-contained library of a single isolated sect copying texts purely for its own internal use. The evidence increasingly suggests that this was a collection drawing on material from multiple places, reflecting a wider network of Jewish scribal activity across the region during the late Second Temple period. That would help explain why the collection contains so many competing versions of the same biblical books, and why texts embodying different theological outlooks sit side by side in the same caves. If people or communities beyond Qumran contributed material to what ended up preserved there, contradictory copies stop being a mystery and start looking like exactly what you would expect from a landscape of active religious debate, rather than a single closed community's settled canon. It is worth being precise about what this research does and does not prove, because it would be easy to overstate the findings. The researchers themselves have been careful to note that DNA evidence sheds light on part of the picture and does not resolve every open question about the scrolls. It has not overturned the idea that a community lived at Qumran or that some scrolls originated there. In fact, genetic clustering among several sheepskin fragments lines up with earlier textual arguments that specific pieces came from local Qumran scribes. What the DNA has done is add a new, physical layer of evidence to a debate that had previously relied almost entirely on handwriting and content analysis, confirming some existing theories, complicating others, and opening entirely new lines of inquiry that simply were not possible before. Consider what this means for how we understand the world Jesus and the earliest rabbis grew up in. The Second Temple period was not a time of one settled, universally accepted version of Jewish scripture. It was a time of active theological argument, competing scribal traditions, and real diversity in how sacred texts were copied, interpreted, and preserved. Different versions of Jeremiah circulating simultaneously, some carried physically from wetter regions into the Judean desert, tell us that ideas and texts moved between communities. People were talking to each other, copying from each other, disagreeing with each other, all while the basic outlines of what would eventually become the biblical canon were still being worked out. That picture of lively, unresolved religious debate is a genuinely different mental image than the old idea of one secluded desert sect quietly hoarding its own private scripture. cut off from the wider Jewish world around it. None of this would have been possible without an unlikely partnership between two fields that rarely interact. A worm geneticist and a biblical scholar, brought together by chance seating at a faculty orientation, ended up pioneering a technique that could read genetic information out of 2,000-year-old parchment without harming a single word written on it. That technique is not finished revealing what it can teach us. Researchers are now expanding the sample set to more fragments, combining genetic analysis with multispectral imaging that can recover faded or damaged text, and building broader databases of ancient animal DNA from across the region in hopes of eventually pinpointing where specific batches of parchment were actually produced. Every new fragment tested adds another data point to a map that is only just beginning to take shape. What began as a shepherd's idle curiosity about a missing goat, and later became a casual conversation on a shuttle bus between two scientists who had never worked together before, has turned into a genuinely new way of reading some of the oldest religious texts in existence, not by translating another word of ancient Hebrew, but by listening to what the animals themselves, or rather their DNA, had quietly recorded about where these scrolls came from and how they moved through the ancient world. 70 years of traditional scholarship built our understanding of the Dead Sea Scrolls, one careful comparison at a time. It took a strand of degraded genetic code, hidden inside sheepskin and cowhide for 2,000 years, to add the next chapter to that story, and there is likely more still waiting to be read.